The Gram stain is used for identifying bacteria as well as staining them. This stain distinguishes bacteria based on their cell wall. Bacterial cells are stained in the Gram stain, and then ethanol is used to attempt to decolorize or remove the stain from the cells. Gram staining is most consistent on cells less than 24 hours old because young cells are less likely to have cell wall damage. Draw 2 dime-sized circles on the back of the slide and label each circle. Prepare a smear. Flame your loop until it is red hot and let it cool. Pass the tube through the flame and use the loop to obtain the broth culture. Pass the tube through the flame again before recapping the tube. Then, spread the culture in the first circle on the slide. Repeat this process so that you have 2 or 3 loops of bacteria on the slide. We will make 2 smears on the slide. Your instructor may have different procedures regarding the number of smears on a slide. Follow the procedures used in your lab. Repeat the process to make a smear in the second circle on the slide. Don't forget to flame the loop and the mouth of the tube between steps. Let the smear air dry. Make sure that the smear is completely dry and then heat fix the smear by passing the slide through the flame 2 or 3 times. Cover the smear with crystal violet for 30 seconds. The exact time for each step will vary with the reagents used in your lab. Wash the slide carefully with distilled water. Tilt the slide and aim the water above the smear. Let the water wash over the smear. Cover the smear with Gram's iodine for 10 seconds to 1 minute. Your instructor will advise you on the required time based on the reagents used in your lab. Wash the slide by letting water wash over the smear. Wash the slide until the water runs clear. To decolorize, let ethanol run through the smear until no large amounts of purple wash out. Be careful not to over-decolorize, as this can rinse the crystal violet from Gram-positive bacteria, giving false results. Wash the slide by letting water wash over the smear. Wash the slide until the water runs clear. Cover the smear with safranin for 30 seconds. Wash the slide with distilled water until the water runs clear. Use a Kim wipe to blot the slide dry. Do not wipe, just gently blot the slide. Examine the slide under the microscope. The alcohol dehydrates the peptidoglycan. Therefore, cells with a thick peptidoglycan wall retain the primary stain and appear purple. These are called Gram-positive. You can also determine cell morphology and arrangement. These are Gram-positive rods in chains. The lipid outer membrane of other bacteria is denatured by the alcohol, and the primary stain easily washes out of these cells, even in the presence of the iodine mordant. The dead, colorless cells would be difficult to see, so they are counterstained to make them visible. They appear red. These are Gram-negative cocci in pairs. When you have finished observing your results, gently blot the oil from the slide and store it in a slide box.